So welcome everybody back. We will talk today about the JAK-STAT pathway. Here's some literature if you're interested in this topic. And now let's dive right into it. We have this receptor subunit 1 and 2. Oh, sorry, there's a mistake. There should be 2. And each of these receptors have a domain which is associated with JAK kinases here. And if a ligand binds on the receptor subunit, this leads to the dimerization of those monomers, which come in close contact here. And then the JAK kinases come in close contact to each other and can phosphorylate each other in crossways, which means this JAK kinase phosphorylates this JAK kinase and just the other way around. The phosphorylation of the JAK kinases then leads to a conformation change, and the JAK kinase can now phosphorylate specific domains on the receptor itself here. And then we have other molecules which are called STAT, and they can now bind to the phosphorylated receptor domain right here. Our STAT molecules have an SH2 domain for binding on the receptor here, and they have a DNA binding domain right here. So by the binding on the receptor domain, the STATs come in close contact with the JAK kinases, and this leads to a phosphorylation of the free STAT ends right here. And because of the phosphorylation, the STATs dimerize at the N-terminus here. And by this dimerization, they experience a conformation change, which leads to the transport in the nucleus right here. The STATs bind on the DNA with their DNA binding domain here. And this leads to the transcription of genes. We have a loop mechanism here. The transcription of those genes leads to the production of an enzyme, the SOX enzyme, which dephosphorylates the receptor here, which resulting in a turnoff of the signal. Let's have a word about the ligands. Those are cytokines. The molecules fold with 4 antiparallel alpha helices that differ in length and straightness. The signal is mostly over the JAK and STAT pathway, and some cytokines react via toll-like receptors. And we have an example for cytokines, for example, the interleukin. The JAK kinases here, there are 4 different types in mammals, and they phosphorylate themselves, crossway, the receptor unit, and the downstream effectors, for example, our STATs. The STATs itself here is a signal transducer and the activator of transcription. STATs associate with the receptor through the SH2 binding domain. They get phosphorized by JAK kinases and they dimerize at the SH2-PY domain. And there's an enzyme which is called importin alpha that transports those STATs, if they dimerize here, to the nucleus. So here we have an cold coil domain, we have the DNA binding domain here, and we have this SH2 domain for binding on the receptor and here for the dimerization. Of course, if we generate a signal, we have to somehow turn it off, and there we have different mechanisms. We can control the activation and half-life of JAKs and STATs. We have this suppressor of cytokine signaling enzyme, which is produced whenever the STATs are activated and they inhibit the JAK kinases. And we have other proteins, for example, the PIAs. This is a protein inhibitor of activated STATs. So this protein will not allow the STATs to bind to the DNA. The JAK-STAT-Signaling pathway has a big role in growth and development of the immune systems, and mutations in this pathway can lead to severe immune, inflammatory or leukemic diseases. I hope this was somehow informational for you. I hope to see you again and have a wonderful day.